Okay. Hello everyone. So again, sorry for the delay. I finally got the problem in my PC. So now we can start. So now I will present my screen. Okay. Can you see the PPT? Is it visible? Hello, hello. Am I audible? Can you see the PPT? Hello.
Yes, sir.
Okay. So let's just quickly start. So in this video, this is the last video of our problem solving session. So in this week 8 video, we will discuss about the previous year assignment of the week 8 lecture of the same course, NPTEL course, cell bio, and the assignment questions will be discussed here, and you will be given a chance to, if you want to guess the answer, and then the answer will be explained, and also some selected topics from the week 8 lecture content will be discussed here. Okay. So let's first start with this question, and before starting we can say that the week 8 lecture content was about different type of cell. It was about stem cell, neuron cell, plant cell, and yeah. So this was the features of the different types of cell. So from this lecture video, the first question is, which of the following tissues don't contain stem cell? Skin, liver, bone marrow, none of the above. So what will be the answer? Does skin tissues contain stem cell? Can anyone guess, please? Hello? Can you hear me? Okay. So Divatmika is saying that yes, skin cell do contain stem cell, right? So which kind of stem cell the skin cell? I mean the skin tissues have. Are they embryonic stem cell or adult stem cell? Can anyone want to comment? Yes. So Divatmika, so your mic is not working. Do you have any problem with mic? Okay, no, okay, so no problem. So you can write the your answer in the chat box, and also others you should actually participate in this discussion. Okay, no problem. So you can write your answer in the chat box, and others, if you, I mean, if it is possible, you can please participate by your audio. Okay, because this is a completely interaction session. Okay, so both of us should interact with each other. So the first question was, and she said that we're following skin cells. Sorry. Yeah, so skin cells contain the stem cell, and which is adult stem cell. What about liver tissue? Does they contain stem cell? Hello? What about the liver tissues? Okay, yes, fine. And what about others? Are you not able to use your mic? Is there any problem in your mic or audio? Someone is asking that how to pass the NPTEL exam. So the main thing is that if you have covered all the lecture contents, okay, if you have covered all the lecture contents and regularly, so you should not face any difficulty in passing the exam. And still I can say that in especially in today's lecture, which is very important for you actually, that you can ask me anything if you have any doubt in this entire syllabus of this course. Okay, so you have got a chance here to ask and clarify your doubts here. So for the last time, uh, because I think it is my last problem solving session. So I request you to ask anything if you have any doubt. And tips for passing exam is just you have to follow the videos. You need to understand the concepts because the questions is most likely to be, you know, very much conceptual. So if you understood the video content, then I don't think there will be any difficulty. Passing the exam is compulsory. Yeah, as much as I know that for obtaining the certificate, you need to pass the exam. Uh, Mridul is asking, doing assignment is sufficient. See, these technical things I actually don't know properly because uh, because my job is only to uh, clarify your doubt regarding your syllabus topics. Okay, so for that I can help you, but in these technical things, I whatever I know I just told you, and uh, for the rest of the things you can contact to the portal contact. They may help you in better way. Okay, because for me I don't know all the things, all the rules and regulations here, but yeah, but for obtaining the certificate I think you need to pass the exam. Yeah, so and don't be scared because exam is will not be very hard. Don't be scared. Just watch the videos, uh, get the concepts, and if you want, I can give you my email ID. So after that session, if you still have any doubt in after reading, so you can just ask me. I will try to respond. And especially in today's class, I request all of you uh, to just clarify your doubts. Okay, whatever you have, don't hesitate to ask. Okay, so yeah, liver tissue also does not contain stem cell. Bone marrow, they yeah, actually, sorry, the skin tissue, liver tissue, bone marrow, all of these tissues contain stem cell. Okay, so here these three options, among these three options there is no option which don't contain stem cell. So for this question, which tissue don't contain stem cell, answer will be none of the above. Okay, let's move on to the question number 2. Which of the following is a type of a stem cell? Totipotent, pluripotent, multipotent, all of the above. What will be the answer?
All of the above.
All of the above. Okay, so what do you mean by totipotent? Especially, I will ask that what is the difference between totipotent and pluripotent? What is totipotent stem cells? Anyone want to guess? Anyone? Okay, email ID I will share. Yeah, so I have shared my email ID. You can take it. Okay, so Ayushi is saying that can. Okay, so I just want to ask all of you that is mic. Is there any problem in using mic? Any technical issue you are facing? Because in other sessions, people use mic for replying or answering. So is it okay? So in chat box, because when I am presenting PPT, it's difficult for me to see the chat box. Okay, let's try that. Someone is saying that ability to produce complete organism. Yes, totipotent is ability to produce. That means totipotent stem cell can be differentiated into any kind of cell. Any kind of cell. So that's why it can give rise to the entire organism. But pluripotent cannot develop. Yeah, pluripotent cannot develop into entire organism. That's true. But what is the main difference? What kind of tissues can be differentiated from this pluripotent, but cannot be differentiated from the pluripotent? That is Pooja Mistry. Yeah. So ask your question, Pooja. You raise your hand. Okay, so the main difference between the totipotent and pluripotent is totipotent. The placenta tissue that can be generated by the totipotent stem cells, but pluripotent stem cell cannot be differentiated into the placenta tissue. So that's why they are not totipotent. They cannot be differentiated to any kind of tissue. Okay, so like they can't be differentiated into the placenta tissue. So that's why they are not totipotent. They are pluripotent. So coming back to the question that yeah, so here actually these three options they are giving here. These three options, totipotent, pluripotent and multipotent, they all of them are feature of the stem cell. I told you the difference between the totipotent and pluripotent, and multipotent stem cell can be differentiated to a particular selected types of specific tissues, not all tissues. So that's why of course they can't be used to develop the entire organism, because they are specific for some particular kinds of tissues. So of course, these three options, toti, pluri, and multi, they all of them are stem cell. So answer is the all of the above. Okay, so the third question is, how do neurons communicate with one another? By electrical events, chemical signaling, radio wave impulse, or electrical event and chemical signaling? Which one is the true?
Via electrical events.
Only electrical events, not chemical?
Both.
Both, right? Very good. So both. Via electrical event as well as chemical. Because because I think, as you know, that some chemicals, name you know, acetylcholine, they are used as a chemical messenger for communicating the neurons. So that's why they communicate with one another with electrical events as well as chemical signaling. Next question is, which of the following is a neurotransmitter? Acetylcholine, serotonin, dopamine, all of the above. What do you think? Okay, acetylcholine is right answer. And what about serotonin and dopamine? Are they neurotransmitter?
Yes.
Yes. Sorry? Yeah, so all of three, yeah, so three of them are neurotransmitter, right? So the answer is all of the above. Yeah. So any doubt till this four questions?
No, sir.
Okay, fine. Fifth question is, which of the following are present in the body's cell wall? So first is beta 1,3 glycan. So is it present? It is present or not? It is present, right? And how these beta 1,3 glucan polysaccharides are formed? Which linkage between the glucose molecules? Which glycosidic bond do you find here?
O-glycosidic bond.
Sorry?
O-glycosidic bond.
Yeah, so the position is beta 1,3, right? C1 of the first glucose and C3 of the second glucose.
Second glucose.
Yes, very good, very good. So that's how this glycosidic linkage is formed, right? So it is present in the cell wall. What about 1,6-glucan? Are they present?
Yes, sir.
Yeah. And how they formed? How the bond formation take place? The positions of glucose carbons. Can you guess?
One glucone having a C1 carbon.
Yes.
And second carbon having a C6 carbon.
C6 carbon. So that's how the beta 1,6 glucan chain are formed. Second is third is N-acetyl glucosamine. So what is the change in the what is the difference between the structure in glucose and N-acetyl glucosamine? Which group is present in the N-acetyl glucosamine? CH3CO energy group is present, right? So that's why acetyl. So this is also present in the budding cell wall. Fourth is manoprotein. Are they present? Yes or no?
Yes.
Yes. So all of these four are present. So that's why the answer will be all of the above. Okay. Yes, answer is the all of the above. Next question is, which of the following nucleus is involved with the mating type switching? So can anyone just tell me in your own language that what is mating type switching? Can anyone guess? Mating type switching, did you go through? Ayushi, what do you think? Mating type switching? What are the two different mating types in budding yeast?
Mat A and Mat alpha.
A and alpha, right? Okay, so Devantika is writing in which cell can switch to locus from. Yes, very good. That is the mating type switching. When the cell will change its mating type, like if it is initially in alpha mating type yeast, then if it will change into A mating type yeast, so that process is known as mating type switching. Okay, so and in the mating type switching, the endonucleus which takes part, that is HO endonucleus, right? Because what is the role of this HO endonucleus? They will make a double strand break. After the double strand break, to repair this double strand break, homologous recombination takes place, and the in the other arm for the A type cell, the A type cell, the HMR alpha mating type will be used as a template for the homologous recombination, and as a result, that mate locus, which is near the centromere, so that mate locus will be converted into the A type from the alpha type. So, and we will discuss about the mating type switching later. So the answer is the HO endonucleus. Any doubt here? Any doubt? Okay, no doubt, so fine. Next question is, just let me. Cells expressing the URA3 gene can be counter-selected with which of the following chemical? FOA, FAA, cannabinoid, 4-FAA and uracil. So, what can be the option? Counter-selectable marker of URA3. Suppose you're transforming cassette, what you are transforming, that contains the URA3 marker. Okay, so now if you want to select those positive transformed cell, you can do the counter selection. So which chemical you will use for this counter selection? Any idea? Any idea you have? Okay, so as you need the explanation for that, the question was suppose you have to transform a transformation cassette which contains the urea-3 marker. Okay, so now if you need to select the positive transformants, then which marker you will use, or which chemical you will use, that is. Okay, so cell expressing the urea-3 gene can be counter selected with which of the following chemicals? What will you use? Do you know the counter selection? Counter selection you know? Okay, so as you know the counter selection, that means for counter selection we are looking for the cells which does not grow in the presence of the counter selection chemical. Okay, like if your cell harboring a urea-3 marker, then if you will use a chemical which will inhibit or which will actually makes toxic product after the urea-3 gene, toxic product due to the expression of the urea-3 gene, so those cells cannot be survived in the presence of that chemical, right? So those are the positive cells. That means they have actually the urea-3 marker in their genome or plasmid, anything. They harbor the urea-3 marker, and that can be selected by using the chemical which, in the presence of the chemical, if the cells harbor the urea-3 marker, that cell cannot grow. That's why it is known as counter selection. But similarly, if you end up use like minus plate, okay, you know when you will plate the transformates in the media lacking the uracil, in that media plate which cells will grow, that will be the your cell positive transformant transformases which contain the urea3 marker. So that is the positive selection or non-counter selection. But the counter selection or the negative selection is you will use a chemical or anything, in the presence of that chemical the cell cannot grow. That means the cell has the marker. Okay. So for urea3, which chemical do you use for counter selection? Anyone remember? For urea3, what do you, yeah, so what can you use for the urea3 counter selection? Very good, 5 of, very good, 5 FOA. Because this fluorotic acid will make a, if the urea3 gene is there and if the urea3 gene is expressed, then the urea3 gene product, in presence of this 5 FOA chemical, it will lead to form a toxic product which is known as fluorouracil. That is toxic for the cell and the cell cannot survive. So if you have the urea3 marker, if your gene is expressing, then in the presence of this chemical the cell will form a toxic material which and as a result the cell cannot grow. So that's why 5 FOA is the counter selectable marker for selecting in the, yeah. So any doubt here? Any doubt here till now? Okay, okay. So move on to the next question. Which of the following is a term for the non-pigmented plastid? Somebody saying something? Yes. Somebody saying something? Okay. Sorry? Leucoplast. Yes, yes. Chloroplast cannot be possible. Chromoplast cannot be possible because all of them are pigmented, right? They have the pigment. Leucoplast, they don't have any pigment, and that's why they look white, right? Like if you see in the radish, there is leucoplast. So as they don't have any pigment, so they are called as leucoplast. Yes. Next question. Carbon fixation reaction, which is also known as dark reaction of photosynthesis, occurs in which portion? Thylakoid. Is it the right option? Thylakoid. Does dark reaction take place in the thylakoid? Can anyone guess? Does it take place or sorry?
A light reaction takes place in the thylakoid.
Very
and
good,
dark
very
in the
good,
stomach.
very good. Light reaction in thylakoid and dark reaction in stroma, right? So the answer will be stroma here. Stroma. Last question is which of the following phytohormones inhibit lateral bud growth? What is lateral bud growth? So which of the following hormone inhibit the lateral bud growth?
Auxins.
Auxin. Yes, auxin. Yeah. Not gibberellin, not cytokinin. It is auxin which prevents the lateral bud growth. So it basically, and what it promotes? What does it promote?
Root elongation.
And stem elongation, right? Yeah. So basically, root elongation is the major contribution of the auxin hormone. Okay. Okay. Yeah. So we are done with the assignment questions. So I will ask you if you have any doubt in this regard. Just then. So try to ask me if you have any doubt. The questions we have discussed, last 10 questions. If you have any doubt, please ask me at any point.
So
Yes.
I had doubt from the ubiquitin proteasome thing. Can I ask that?
Yeah, sure.
From Olio.
Yes, sure.
Can you please explain that pre-RC complex in that?
Sorry?
Pre-RC complex.
That is the. Yeah, so that is in case of replication, right?
Yes, I think so.
Replication, right? Ha. Pre-RC.
Yes.
Yeah. So it proteasome ubiquitin. What doubt do you have?
I didn't understand that role of that pre-RC complex. Like, how does it work in the entire system?
Pre-RC complex, right?
Yes.
Okay, so let me just. Okay, let me open another PPT. So so if you are, can you see the slide, the image?
Yes.
Okay. So in this case, what is happening that for starting the replication, the origin of replication should be fired, right? From that, the replication will be initiated. So initially, what happened? The ORC proteins and CDC6 they bind to the origin of replication. Okay, and when there is any need of replication right in the beginning of the S phase, then this helicase will be recruited here, and after recruitment the replication will be resumed. So here you can see that
The
in,
one second. The
ye-
CD1 and the helicase is together, and then they are broken apart for
yes,
that
yes,
RCA to
yes,
form.
because
Okay.
for loading the helicase you need a CDT1, and the CDC6 which is initially bound to the ORC, and ORC is bound to the original replication. So this CDC6 complex is playing a role of having a landing platform for this helicase and CDT1. Okay, it will actually give a landing platform for this helicase. So due to this, helicase can be loaded here by the help of CDT1, and then both of this CDT1 and CDT6 will be dissociated, and after this, what will be left out here, the ORC and the MCM helicase, that is known as PRC complex, pre-replicative complex. And after that, when there is a upregulated level of SCDK in the initiation of the S phase, so now SCDK will actually try initiate the replication process. Okay, so now do you have any idea of
I
what
had a doubt. I had a doubt if the pre-RC is made up of ORC and two
huh,
MCM.
huh.
Where is that second MCM coming from? I didn't understand that.
Second MCM, there is actually two copy of see, there is actually, although in the figure it is showing that there is only one MCM helicase, but you need two MCM helicase, because one MCM helicase, see in this figure, one MCM helicase will go in this direction, and another MCM helicase will go in this direction. This MCM helicase will use this strand as a template, and this MCM helicase will use this lower strand as a template. So for that you need two MCM helicase, although there is only one is showing here, but the loading process is same. Over the CDC6 they will be loaded by the help of CDT1. So that's why you can see here two copy of MCM helicase is required to be loaded here. Okay, clear?
Okay. Yes.
Yeah, because one is in this direction, one is in this direction. That's why two copy. Clear? Okay, and you told me that you have some.
Yes. Okay.
Okay, so and you told me that you have some doubt in ubiquitin system. What is that? Or it was the only doubt of you?
This was only the doubt.
Okay, fine. Okay. So any more doubt here?
So just 2 minutes. Can you give me? I'll just check.
Sure,
If I have any more.
Yeah, sure. Actually, all of you can do this. You can just take your time, and you can just let me know that whether if you have any doubt or not. I will let you resolve. Take your time, and let me know. After that, we will shift to the second part of the session. Okay, so one question I got till now, that MRC mutant, right? MRC mutant is just, MRC protein, right? MRC1. I think this slide also has this. Just a minute. Yes. So, do you know MRC1? Ayushi, who was asked the question, the role of MRC1. So the role of MRC1 is, when there is any DNA lesion, DNA damage, then replication fork will be stopped there, right? It will be stopped here. So after this stopping, there will be creation of this single-stranded DNA. And as we know that if there is any single-stranded DNA exposed, then that will be bound by the replication protein A, RPA, which is also known as SSB, single-stranded DNA binding protein. So this RPA foci, when it will be created here, that will be recognized by the MEK1 protein, which is the component of the S-phase checkpoint pathway. So the MEK1 protein, by this GBC to domain, it can recognize this RPA multifoci. And after recognizing this, MEK1 will be activated, and it will be activate its downstream activators. So one of them is MRC1. MRC1 is phosphorylated by the MEK1, and it leads to the phosphorylation of the RAD53. And after the RAD53 phosphorylation, the different steps of fixing this damage will be taken place, like it will inhibit the mitosis, it will inhibit the homologous recombination, it will induce the expression of the genes, especially the expression of the DNA repair genes, and it will lead to recruit the translation DNA polymerase by so this and also it will inhibit the late replication origin firing, because until and unless this damage will be cured, that origin should not be fired. So that will be taken care by this S-phase checkpoint complex. So that is the MRC1, and when you mutate the MRC1, then your S-phase checkpoint complex will be compromised. So especially if you will grow the cell in a replication sensitive drug like hydroxy-MMS, then your as your MRC1 is mutated, so your S-phase checkpoint complex will be mutated, non-functional. So there will be no functional checkpoint, and the cell will not be arrested here, and it will be just go on without fixing the error. So that will result into the seeker phenotype in the cell. They will grow less. So now, so now is it clear? MRC1 mutants. And any more question you have? Tell me. I will try to. So for important topics, as I already told that every topic is important here. here. So actually, it is difficult to filter out some topics, but there is no such kind of, because question can come to from anywhere, and the main thing that you should make it clear that for every lecture you should have a clear concept. If you have this, then you should not. So if you have this, if you have a clear concept, then you should not face any kind of difficulty. So among them also important topics which can be made like S phase checkpoint, M phase checkpoint, ubiquitin proteasome system, you know. So this kind of topics, M phase checkpoint, and also about aging, apoptosis, which we discussed in the last class. They are actually very hot topic, but in spite of that, every topic is important. You should know, you should actually have a good concept about the every topic. Okay, so uh, there is no such topics, important topics to be focused particularly. Just build your concept, everything will be easy in the exam. Okay. Okay, any more doubt? Anyone have any more doubt? You can ask. Okay, so the histone tells. Okay, so as you know that the histone code is what? Histone code is composed of octamer histone code is composed of two copy of each of H2A, H2B, H3 and H4, right? Right. So two copy of this will form a octameric histone or nucleosome, and DNA becomes wrapped around this nucleosome or this histone core subunit. Okay, and apart from that, each of these subunit H2A, H2B, H3 and H4, their intermediate tail are not a part of a code component of the histone. So they are actually protrude from the that histone code subunit. Okay, so they are projected outwards and they are known to interact the nucleosomal DNA, and they are yeah. So the main role of this histone tail is, which has been shown, that the main role is to stabilize the histone octamer, the histone core complex H2, H2B, H3, H4. This histone core complex is stabilized by the histone tail of each of these four subunits. So that is the histone tail. Are you so did it clear your doubt? Okay, any more question? Anyone? Any more doubt you have? Okay, so let's move on to the our next part of the lecture, where we will discuss some selected topics. Okay. So here first we will discuss about the mating of the yeast. So as you know that there are two kind of haploid budding yeast is there, A type and alpha type. I think someone is in the queue. Okay, so there are two types of mating type of the haploid yeast. One is A, one is alpha. So this A type of budding yeast, they have a receptor for the alpha, for the budding type alpha here in the cell membrane, and the alpha type cell has a receptor for the mating type A. Okay, so during mating, what will happen? This A type cell will secrete the mating factor A, that is the blue color, and uh, in the vicinity, the alpha mating type cell will secrete the alpha factor, which is colored in red. So after this secretion, this A type will receive the alpha factor and alpha type will receive the A factor. After that, when A type will receive the alpha factor and alpha type will receive the A factor, then their cell the cell cycle will be the cell division will be inhibited, and then they will be in the interface. They will not be divided. So after this, after receiving this mating pheromones, there will be a change in the morphology of the A type cell and the alpha type cell, and after some time they will be fused to make a diploid cell A and alpha, which and this diploid after the mating, this diploid body yeast will contain both mating type alpha and mating type A. That's what in one chromosome it will contain the mating type A, in another chromosome it will contain the mating type alpha. So that's how the mating of after the mating of budding yeast, there is a diploid budding yeast will be formed. Is it clear or not? Mating of yeast. Okay. Okay, so the next is how this, when the cell will receive the pheromones of mating type, how this will lead to a signal transduction in the mating yeast. Okay, so for that, when this pheromone will be received by a GPCR, and in budding yeast there is actually state 2 and state 3. They are the receptor for this mating type factor pheromones. So this state 2 and state 3 will receive which are a GPCR. So after the receiving, GPCR will be activated and G alpha will be dissociated from the G beta gamma, and this G beta gamma, what it will do, it will first, after the activation, stage 5 will be recruited to here, and after the stage 5 recruitment, then stage 7 will come here, which is a MAP kinase, and fuse 3 will come here, which is a MAP kinase. So MAP kinase, as you know, it will phosphorylate the MAP kinase, which is fuse 3 here. Okay. So this, actually this stage 5, when it will recruit to here, this stage 5 is a MAP kinase. So it will phosphorylate the stage 7, which is a MAP kinase, and this stage 7 will phosphorylate fuse 3, which is a MAP kinase, and this will be transported into the nucleus, and in nucleus it will phosphorylate the DIG1 and DIG2 protein, which will lead to the dissociation of the DIG proteins from the state 12, and this stage 12, after that it will be activated and it will be act as a transcription factor, and it will induce transcription of several genes. So that's why, and these genes, one of the so these genes are known as mating response gene, which is required for the mating of two different mating type cells. So I will be repeating again. I think some so I will be repeating again. Pheromones will be received by the GPCR. GPCR will be activated. G alpha will be separated from the G beta gamma. In G beta gamma, first stage 5 will come. in the activated G beta gamma, which is a MAP kinase, it will phosphorylate the stage 7, which is a MAP kinase. Stage 7 will phosphorylate FOS3, which is a MAP kinase. The phosphorylated massive will come into the nucleus. It will phosphorylate the DIG proteins, and these DIG proteins will be dissociated after the phosphorylation, so that the state 12 will be free, and this free state 12 will act as a transcription factor to induce transcription of the mating time response genes. Okay, so that's why the gene signal transaction of the mating in the mating yeast. So, any doubt here? Okay, so no doubt there, right? So let's move on. So that is the mating type switching. As we discussed about the mating type switching. So how is it happened? This is the organization of the chromosome 3 of the A cell, and this is the alpha cell. In chromosome 3 there is one mating type locus, mat locus, which may be A for A cell and alpha for alpha cell. And in addition to that, there is two transcriptionally silent region of mating type. One is HML alpha and another one is HMRA. That is same for the A cell as well as the alpha cell. So the difference created between these two A and alpha in the bad type locus, A or alpha, which is with the true. Okay, so this is the chromosome 3 organization of the A cell and alpha cell. So during the mating type switching, in this conformation you can see that there is a, this is the, if you will compare with this, there is a HML alpha, then MAT A, then HMRA. So when it needs to switch its mating type, then it will fold like this, and here there is a HO endonuclease site where the HO endonuclease make a demonstrative break here. Okay, in the MAT A locus for the A cell. So in MAT A locus, when it will make this DSB demonstrative break here, so after that homologous recombination will be taken place, and this will use this HML alpha as a template to copy this region. So as a result, after this homologous recombination, this bent locus from A, it will be converted into the alpha, because it copied this HML alpha, which is a alpha. Due to this, the bent A will be converted into bent alpha. So that's how, and this is the more precise way how this DSB repair takes place. In mat A, when this DSG will be created by HO, this will be the case, and then it will be homologous because this X and Z of mat is homologous to the X and Z of HML alpha. See, X and Z of HML alpha is homologous to the X and Z of mat A. So thus this homologous recombination can be occurred, and as this will be acted as a template, then this this will copy this entire region. So this YA will be copied here in the mat locus. So as a result, there will be a actually this HML alpha will be copied here. So here alpha will be copied. So this YA will be converted into the Y alpha in the mat locus. So that's why the A cell to alpha cell beta type switching is taken place. Okay. So yeah, so any doubt here? Let me know. Any doubt here? Let me know. Okay. So next we will see that we will discuss some technique about the epitope tagging. So if you want to tag in the C terminus of any particular ORF, so for this epitope tagging, what will we do? We will use a plasmid which contains this tag and the corresponding marker. So we will design a primer which has the complement sequence here, and this region. Yeah. So after this, we will set up PCR by using these two templates. We will copy this region, and as the primer contains homologous region of this, so this region will be copied here, and this RP contains the homologous region of this. So this region will be copied here. So this is our PCR product that will be transformed into the yeast cell. So after the transformation, as just see it here, as this has a homologous recombination, homologous region with this, and this has a this. So homologous recombination can be occurred, and after the homologous recombination, there will be in the C terminal there will be tag, and the selection marker will be there. Okay. So that's why we do the C terminus epitope tagging by PCR mediated method, because here we will amplify the cassette which contains the tag and marker by PCR here, and the primers, as the primer contain the homologous region of this, so the cassette will also contain the homologous region, and this homologous region will make a homologous recombination here, which will lead to the insertion of the tag and selection marker at the C terminal end of the yeast genome. So that's why this particular WRF of interest will be converted into the actually it will be tagged by this tag and this selection marker, because if you will transform this, you need to know that to screen for the positive transformant you need some selection marker so that you can select your transformant. So that is the method of PCR mediated epitope tagging. Any doubt here? Just let me know. Any doubt, please let me know. Okay, so let's move on to the next yeah. So now for the deletion thing, if you want to delete a essential gene, then you cannot delete the essential gene directly, right? Because if you will directly delete the essential gene, then what will happen? The cell will be very sick and the cell cannot grow. because the gene is essential for the survival of the cell. So for that, we follow a process called plasmid shuffling. So here you can see from this diagram that in a W3O3-1A, which is a haploid yeast strain, A type haploid yeast strain. So in this yeast strain, if you want to delete a particular like NAC3 gene, okay, NAC3 is a component of the SMC56 complex. So if you want to delete this NAC3 gene, which is an essential gene from this haploid budding strain, so for that we cannot directly delete this, right? So this is the genomic DNA of the W3O3 where NAC3 is present. So before deleting this NAC3 from this region, we will transform our plasmid which carries the wild type NAC3 copy and which is a uracil marker into this W3O3-1 strain. So after the transformation, the strain will have a NAC3 in the genomic DNA as well as it will also have a plasmid which contains the NAC3 wild type copy with the uracil selection marker. After that, we can transform a NAC3 delta cassette with a selection marker of kanamycin, resistance gene. So when this cassette will be transformed here, so as you know that this is the cassette and this is the genomic DNA. So now you can find the homologous region here and here with the genomic DNA portion. So that will be undergone the homologous recombination, and after the homologous recombination, the genomic DNA, the NAC3 will be deleted and it will be disrupted by the kanamycin resistant marker. Okay, and also in addition to that, the yeast strain have a plasmid carrying the NAC3 wild type copy. Okay. So now the yeast strain has the deletion of NAC3 here in the genomic DNA region, but it also has a wild type copy in the plasmid. So although it has a deleted copy of NAC3 here, NAC3 is deleted from the endogenous locus, but till it has a NAC3 wild type copy in the plasmid, right? So due to the presence of the plasmid, the cell will not be sick because it has the wild type copy in the plasmid. So now there is two possibilities here. Either the homologous recombination will be between the cassette and the genomic DNA, then this thing will be happen, or the homologous recombination can be possible by the cassette and the plasmid, because homologous region is also present in the plasmid, right? Because it contains the NAC3 wild type copy. So if this happens, then this situation will be happen. There will be the NAC3 wild type copy in the endogenous locus of the genomic DNA, but the plasmid will carry the NAC3 deleted copy. But we don't want that. We want deletion from the genomic DNA region, right? So this thing, now for imagine this cell, if you will grow in SCFOA, then in FOA it will not grow because it has a uracil marker here, uracil protein. Now as it does not have a wild type copy in the genomic DNA, so as it is an essential gene, so it need to maintain this plasmid, because this plasmid only contains the necessary wild type copy. So this can grow in the SC minus ura media, but it cannot grow in the SCFOA media because uracil is there, because its plasmid is maintained. But in this case, as the genomic DNA contains already the NAC3 wild type copy, so it does not need to maintain this plasmid. Okay, and if you will grow it in the FOA, then of course it will kick out the plasmid, because uracil will make a toxic product in the presence of FOA. So it will kick out the plasmid from the cell, because to just prevent the formation of the toxic metabolite, because it has the NAC3 wild type genomic DNA. So it already has. So it does not need to maintain the plasmid. So it can grow in the SCFOA. So that's how we can differentiate that the cell which does not grow in the SCFOA, that is our required strain, but the cells which grow in the SCFOA, that is not our required strain. So that is the way of plasmid shuffling, where we can delete the essential gene. So any doubt here? Okay, so let's move on to the stem cell portion. Okay, so the stem cell portion, uh, so initially Dr. Gordon discovered one thing, that he was able to, you know, what he did from the frog A, he took out the egg, and to get rid of the nucleus, he did the irradiation of the egg, so the nucleus will be non-functional, and this will be form a enucleated egg without a nucleus. And from the frog B, he collected the skin cell. From the skin cell, he isolated the nucleus, and he mildly injected the nucleus into this enucleated egg cytoplasm. So after this, as this cytoplasm of the frog B egg, it contains the cytoplasm of the frog A egg, there is a nucleus of the frog B, so it will create the clone of frog B. So that's how he so that's how he showed everyone that from an egg of a frog A, by using the egg of a frog A, you can have a clone of frog B by fusing or microinjecting the nucleus of frog B. And after that, Yamanaka discovery came to take place, where they from the mouse they collected the fibroblast tissue. In the fibroblast tissue, they used 4 transcription factor: OCT4, SOX2, KLF1, and CMYC, and this transcription factor will lead to the de-differentiation of the fibroblast, and it will make the fibroblast to the induced pluripotent stem cell, from a differentiated cell to a stem cell. So it was just opposite, reverse. So that was possible by using this transcription factor, transfecting these transcription factors. So now, what is the use of that? Now, in the from this iPS induced pluripotent stem cell, you can use the if you want to develop a osteoblast here or a neuroblast here from this stem cell, you can use that particular tissue specific growth factor hormones. Then you will get the osteoblast, you will get a neuroblast, you will get a chondroblast. So main thing is that if we can go back to the stem cell, then by using the stimuli hormone growth factor, you can develop any kind of tissue you want. So that is the different very important thing is here, that why your iPS is required. Like for example, a patient of accident, so if he needs a so if his heart is damaged, so then the doctor what can do, they can cut a tissue from other section like skin, like fibroblast. So they can take the fibroblast tissue of that person of that patient, and they make the iPSC from that, and from the iPSC they can develop, or they can differentiate that iPSC to the heart tissue, right? So that's how they can actually regenerate the heart tissue for any case of emergency. So that is the induced pluripotent stem cell concept. Okay, so any doubt here? Okay, so let's move on to the next topic. Now, what is the application of the iPSC? So if from a patient you can take a somatic cell, you can add those transcription factors and you can get the induced pluripotent stem cell, and then you can make the differentiation of your own choice. If you want to give rise of cardiomyocyte, you can use that specific hormone stimuli growth factor. Similarly, you can from this stem cell you can get hematopoietic cell, islet cell which is in the pancreas, you can get the neural cells, and then after getting this, like for example, if the patient needs the cardiomyocyte tissue, then from the somatic cell you can make the iPSC. From the iPSC you can differentiate the iPSC to the cardiomyocyte by using the specific growth factors, and then you can transplant this cardiomyocyte to the patient. So by using the somatic cell, you can get the cardiomyocyte. So that is the beauty of this iPSC, that you can, once you get this iPSC, you can you may be able to differentiate into the any tissue which will be required for the transplantation, right? Yeah, so how iPSC in disease modeling, how it is useful. So you can see that here uh, suppose a patient has a problem in a neuron, okay. So yeah, so for that you can do one thing. You can take the patient somatic cell. From the somatic cell you can make the iPSC by using those 4 transcription factor and you can use the CRISPR-Cas9 to edit the whatever problem in the genetic sequence, and after that editing you can get the genetically corrected iPSC. Now this genetically corrected iPSC you will take and you will differentiate it into the neuron which will be the correct neuron. But from the but if you will not do this CRISPR-Cas9 by mediated gene editing, then your mutation will be evident here. Mutation will be there. So this iPSC, from this iPSC we will develop a neuron that will be mutated neuron or mutation carrying neuron. So by this process you can by using both iPSC and the CRISPR-Cas9 process, you can genetically edit the problem in the cell and you can get a correct neuronal tissue. So that is important here, that how your disease can be alleviated by using these two techniques. Now we can discuss about some plant cell morphology. In plant cell there is like animal cell, there is nucleus, there is endoplasmic reticulum, Golgi body, mitochondrion. But the difference is the vacuoles they have, they are more in number and they are actually more in size in respect to the animal cell. Also they has a unique thing that is called a chloroplast, where the light reaction and the dark reaction of the photosynthesis is taken place. So that is a different thing which is unique for the plant cell, this chloroplast and the vacuole. Yeah, so just remember that. And plant cell mitosis. So from the initiation, yeah, so for the plant cell mitosis, so it will so let's discuss about the plant cell mitosis. So it starts from the interphase where the chromosomes are decondensed. So after that you can find a P-prophase band that is generated by the polymerization of the microtubules. This P-prophase band will be formed, and after that it will transit to prophase when the chromosomes will be duplicated and it will be condensed. Here in the nuclear envelope you can see that will start disappearing, and after prophase it will go to metaphase. In metaphase you can see the chromosomes will be aligned in the plane of the cell division, and the spindle fiber will be originated and it will be connected to the kinetochore of the of these chromosomes. After that anaphase will be started. In anaphase these histochromates will be separated to the opposite poles, and after the separation completion the cell plate will be generated in the position of the that cell division plane, and in the late telophase there will be nuclear envelope the nuclear so in the late telophase the nuclear envelope will come back, the cell plate formation will be complete. Now the cell can be divided and it will again go to the interphase and then again the same thing. This is the plant cell mitosis process. Anyone any doubt here? Let me know if you have any doubt. Okay, so last we will discuss about the oxygen inducible degrowth system. So it is useful when we need a cell cycle, if we need a cell cycle specific deletion of a protein, but we need to present the protein in other phase. Like if I want a protein only to be deleted in the S phase but not in the G1 phase. So for this cell cycle specific deletion of the protein, we need a oxygen inducible degron AID tag of a protein of interest. So here in the POI, a AID tag was there in the okay. This AID tag, what is the role of this AID tag? That is oxygen inducible degron tag. That there is a ethylbutyl ligase where there is a Cul1 RBX1 which is a E2 binding site, and SKP1 bound with the TIR1. So when you will feed the oxygen, then the oxygen will interact with the TIR1 and with the AID. So as a result, it will play a role of the mediator of a bridge of the mediator of or a bridge between this ethylbutyl ligase and this protein of interest, which is AID tag. So now this substrate will come to close proximity to this ethylbutyl ligase, and this ethylbutyl ligase will make a polyubiquitin chain formation to this protein of interest, and it will be degraded by the proteasomal system. So here the thing is that even if we don't add the oxygen, then there will be no mediator between this ethylbutyl ligase and the POI. So this substrate will not come to the close proximity of this ethylbutyl ligase and no degradation. But whenever you will feed with oxygen, then oxygen will make a bridge. It will interact with TIR1 as well as it will interact with the AID, and due to this interaction, now this POI will be come to the close proximity of this ethylbutyl ligase, and then this E2 will be recruited here, and this it will transfer the ubiquitin, and the polyubiquitin chain formation will be happen. After that, this polyubiquitin related, the POI will be recognized by the proteasome, and in the core subunit of the proteasome, the protein of interest will be degraded. So that's how the oxygen inducible degradation system is working, only in the presence of oxygen. So if you want the protein degradation and only in the S phase, then only in the S phase you will add the oxygen, and after the S phase you will remove the oxygen. So as a result, only in the S phase, your protein will be deleted. Your protein will be deleted only in the presence of the S phase. And whenever you will wash the oxygen after the S phase completion, then again the protein will be started expressing. So that's why if you want a cell cycle specific deletion of a protein, then you use this oxygen inducible degradation system here. So now let me know if you have any doubt so far. হয়ে গেছে। তুমি কোথায়? শরীর খারাপ? এটা পছন্দ হয়নি? রাখলাম না বল। আমি কি? আমি ওটা কিনে দেয় কোথায় ওটা? তো কে বলো কিনে দেবে? দেখ দেখ 1500 কত? 15। বাহ চুপ বাহ বাহ তার 1500 টাকা কি? আই পছন্দ হয়েছে কি হয়নি? বাহ 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 আমি তোমাকে দিলাম আর তুমি আমাকে দিয়ে দেবে বাহ তুমি খেয়েছো কিছু ওটা দেখে পিস্তা পিস্তাও তো আছে ওর মধ্যে পিস্তাও তো আছে বলছি ওর মধ্যে পিস্তাও আছে ঠিক আছে তুমি ওটা তোমাকে তো একবারে খেতে বলছো না তুমি তোমার কাছে রাখো পিস্তাটা তখন খেয়ে That is really bad. Why? তুমি সবকিছু আমাকে দিয়ে দাও কেন? আচ্ছা হ্যাঁ কি হ্যাঁ 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 ওই জন্য ঘুম থেকে হ্যাঁ ঘুম থেকে উঠে আমাকে যাবো ঠিক আছে আচ্ছা Bye. বলেছে কি মনে রাখো একসঙ্গে খাবো একদিন একটা তখন ইচ্ছা প্রথম ইচ্ছা হবে তখন ইচ্ছা হবে তখন খাবো একদিন একটা খেয়ে গেলে একটা খাবে খাবে যদি চারটে করে খাবে যদি কাউকে দিতে ইচ্ছা হয় কাউকে দেবে এটা এমন কিছু নেই যে এত বড় রোজ একটা করে মুভি দেখেছি তার সাথে ভালো লাগছে একা ভালো লাগছে না সত্যি লাগছে বলতে কোনো ইচ্ছে করছে
बीचमध्ये बोललो तुला जाना अच्छा तरकीब नक्की की होती